Hi, everybody. Thank you for bearing with us. We've still got people joining, so we're just going to wait a moment so that people can connect with us. Okay, welcome everybody to the Sequence ME and Long COVID webinar. We're so excited to be with you today. We've got an opportunity for you to hear from several of us about the study and the progress that we've made. And Sequence ME and Long COVID is a partnership between Action for ME, Edinburgh University, Embol EBI, Oxford Nanopore Technologies, and most crucially, people with lived experience. It's a Decode ME study, and we're building on all the work that we've undertaken and the participation and engagement that we've had through that study to be able to develop our understanding from a genetic perspective into ME chronic fatigue syndrome, but also long COVID. You'll hear about the science behind the long read whole genome sequencing and also why it's a step up from previous approaches. And we'll give you a quick overview of the phases of the study, some of which have some funding and we've made a start, and others that have yet to come a bit further on. You'll also hear from Professor Chris Ponting, who will talk about what the findings could mean for diagnosis, treatment, and further research. So I'm now going to hand over to Chris. There will be time for questions and answers at the end. There is a Q&A function, so please type your questions in there. We might answer some questions live as they come in. Some of the team are working in the background. Just a little reminder, please don't share any personal information. We can't answer any questions about your background, your experiences. We can only talk about the work that we're doing in have done in Decode ME and Sequence ME and long COVID. So I'll now hand over to Chris.
Thank you, Sonya. Next slide. So we're going to talk today about the new project, Sequence ME and Long COVID, which, as Sonya has said, is a Decode ME project, and it works under its ethics and its ethics approval. It's a multi-phase project, so we'll discuss today the first two phases, and I'll be covering mostly phase two. We're going to have hopefully quite a long time to have a Q&A session. I'm very happy to answer what questions we can. Next slide. So how we got here, of course, it's a long journey. When we initially designed Decode ME, we realized that there was always going to be an opportunity to do whole genome sequencing. And today we are discussing what whole genome sequencing is. And I'm happy that we made that decision all those years ago because that then made this a possibility. Essentially, these are the same objectives as before. As for Decode ME, we want to find out the genetic differences between people with ME and the general population to find the biological mechanisms behind the disease, because only then will we know what are the targets of genes that we can take to the pharmaceutical industry and get them to follow up on. So we have the fantastic contributions made by thousands of people across the UK who are above the age of 16, have a medical diagnosis, and their data, your data, have contributed to the Decode ME results that we pre-printed last August and still are working on. The important thing, and we always need to say this, is that the Decode ME study and in the future the Sequence ME and Long COVID study will benefit enormously from people with lived experience. So the Decode ME project, as we've said, has recruited thousands of people and have generated these 8 genetic signals that are genome-wide significant. So we're still working on this and we'll report as soon as we can. This is therefore a different approach. And in the next slide, I'll show what we have, if we can, Sonya. Thank you. So one before that, that there is a limitation to what Decoder Me could ever have found. And that is, we could not pinpoint a single gene because there was and is always the possibility that the signals we're finding point to a neighboring gene rather than the one that we're identifying as being the most important. So to get around that ambiguity, we're using this different technique, and that is whole genome sequencing. And on the right, and this slide will show you the differences between the two methods. First of all, as the name implies, it sequences. It reads out every single one of the 3 billion letters that is in your genome and mine, as opposed to in Decoder Me, 1 million letters or so for the genotyping array-based method. So because we're able in sequencing to read out all of the letters, it can find things that Decoder Me could not. In particular, rare variants. These are often private to individual families, are therefore possibly going to be those that are found to be explaining people's symptoms. So whole genome sequencing finds everything, but it's more costly. That's why we didn't use it in the first place. And the data that comes out of it is huge relative to what we have already produced using Decoder Me. There is something called structural variation, which is not about a single letter, but about a tranche of letter, a portion of the chromosome. And that will be newly visible in this approach, whereas for Decoder Me, it was entirely invisible. And I'll explain that later. So the questions asked are different. Sequence ME and long COVID's question is, well, what are the ME/CFS genes? Previously in Decoder Me, we asked a different set of questions. We asked, is ME genetic? And the answer was in part yes. And we asked, what bodily system is the genetics most focused on? And the answer was neurology. So with these two different methods, and we'll discuss this in a minute, next slide, we'll find out that they're complementary. But the key analogy that I think I want to leave you with is that whole genome sequencing is like a very focused spotlight, whereas what we used for Decoder Me was a sort of a floodlight. And so the whole genome sequencing can illuminate genetics further and are able to see things that you can't see in the Decoder Me approach. Next slide. So more specifically, what whole genome sequencing does is to pinpoint genes that are primarily affecting ME and have little effect on other traits. And this paper on the bottom right gives the evidence for that in general. Whereas if you click again, the whole genome sequence approach, the GWAS approach, looks and finds intervals that contain several genes that most often affect other traits. In other words, as well as ME/CFS. So it's much more fuzzy than is whole genome sequencing, and yet they're complementary. So by having both, we're going to be able to have a fuller picture of all of the genetics that we can find given the numbers of people that we have. Okay, so just to remind you why we're doing genetics in the first place, and that is because the pharmaceutical industry is very keen on genetic evidence, and that's because it improves their ability to produce effective drugs. And because the production of drugs and the whole drug development pipeline is incredibly expensive, what we need to do is to have the genetic evidence that's very compelling to go to the pharmaceutical industry and say, here are the genetics. It's now up to you to do what you can do to generate the drugs. So that's why we do genetics. So how are we going to analyze the data in time? And this is fairly complicated, but let me step through it one step at a time. So this is the whole genome sequencing approach. And what we're looking for in each person, and in pink is the person with ME, and in the blue is the person who's a control individual, we find the letter differences and we call them, we find them. And then among all of those, we work out which we believe to be damaging that break the gene and its function. And once we've done that for all of the cases, we collapse all of the damaging variants and count them and see whether essentially there are more of those variants than in the controls. And we do a statistical test, and that produces a probability value, a p-value per gene. And then we have to realize that because so many tests have been employed, we have to use a quite stringent threshold. And if that threshold is achieved, then this is a gene that becomes an ME gene that has been broken more often in people with ME than in controls, but often in different ways. Whereas in Decode ME, we were finding the same variants being different on average, more in people with ME than in controls. Next slide. So this is what others have done thus far. This is AstraZeneca as a company. They did this analysis, and I think the data here shows for about 2,500 cases and almost 500,000, 122,000 controls. And the gene that is most significant is a gene that is in one of our decoder me intervals, BTN2A1, which is therefore of interest. But you'll see that none of these genes achieves the significance threshold used in this study. Now, for a single disorder like ME, what we need is essentially these data points to be slightly higher than the one for BTN2A1. And we will achieve significance in this single trait study. And we believe that by having 6,000 cases, which is our intent for this year, we will be able to achieve that. Next slide. I said to you that we're not just looking at single letter changes, we're looking at structural changes in the genome. There are a whole variety of different types, as in the bottom right. And I'm highlighting here one for a study of not ME/CFS, but chronic fatigue, where there was, for a single individual, the discovery of an inversion in these two genes, which affects their, or potentially affects their function. So these are the kinds of things that we would see with the long read technology we're using, and actually are not seen both in the decoder me type technology and in shorter read whole genome sequencing approaches. So next are the different phases. So I'll run through these quickly and then hand back to Sonya. So Sonya will explain phase 1 in a minute, and I've just told you about what we're doing for phase 2. 6,000 ME/CFS samples. We've recruited those already. We have those in our freezer here in Edinburgh. And in the future, what we do next is to analyze the data and compare to existing control data. In future, we will look at whole genome sequencing of 3,000 more ME samples, and then move on to the long COVID portion of the study, where ultimately we will sequence 9,000 long COVID individuals. And we estimate that the entire project requires about £20 million. Sonya.
Thanks, Chris. So as Chris mentioned, I'm going to tell you a little bit more about phase 1. Each of the phases are likely to be led by different delivery teams. And so phase 1 is going to be led by Action for ME. Phase 2 that Chris has just shared with you is being led by the University of Edinburgh. So phase 1 is very much about getting the project going. And what we have been looking at is to put all the foundations in place to be able to get the study up and running. As I mentioned earlier on, this is a Decode ME study. So the Decode ME Management Group, that's myself, Chris and Andy Devereaux-Cook, will continue to oversee and lead the study, but we are looking at potentially expanding the group. We've been setting up the governance. We've been looking at what information we need to share at this stage. And like Decode ME, we will gradually build that up as the study progresses. So if you want to find out information and read the frequently asked questions, then you can go to Action for ME's website, and there's information on there specifically about Sequence ME and long COVID. The other thing that we are doing is to look at the fundraising for the remainder of the study. We've been successful in securing funding for phase 2, but there's a lot more to be done, as Chris shared. And we need to be able to set up strategic partnerships, look at investments to enable us to be able to deliver that. And at the heart of all of that, of course, is making sure that people with lived experience of both ME and long COVID help drive and deliver the study in the way that we did with Decode ME. We know that the science is better as a result of the involvement of people with lived experience, and we are all fully committed to making sure that that is pivotal to Sequence ME and long COVID. One of the things that we will be setting out to deliver in phase 1 is to establish the recruitment criteria for the long COVID samples. As Chris mentioned, we've already got a large number of samples within the Decode ME cohort, and we will be analysing 6,000 of those with the funding that we've already got in phase 2, and we want to do another 3,000 thereafter. But we have yet to recruit the participants with long COVID, and we need to establish what criteria we are going to use to do that. We went through a very similar process with Decode ME. We worked with a large number of people with lived experience and scientists and others to really refine the criteria, and we will follow exactly the same process during this phase to be able to determine the recruitment criteria for long COVID. So the other thing that we're going to be doing is gathering expressions of interest. Again, we did this with Decode ME whilst we were working out the criteria we were using. We will ask people to sign up, and we will then recontact people when we're ready to start recruitment. But we're not going to be recruiting during phase 1. And then the other thing, as I mentioned, is the strategic partnership development. And that crucially is to establish funds. When we set out the project plan for phase 1, we set out to raise, I think it was something like £6 million within phase 1 to enable us to be able to take the next step and start the sequencing. We've actually already achieved that, and having just brought in a new colleague to assist with the fundraising, we are now hopeful that we can go ahead and fundraise for the remainder of this study. So the funding for work package 1 is just over £350,000, and we have funding from a diverse range of sources. We were delighted to receive our initial piece of funding from the Schmidt Initiative for Long COVID, or SILK, and we've since had funds from CODA, Complex Disorders Alliance, and Solve ME in the States. There's also funds that have come in through private donors and Action for ME. And we've got about £30,000 still left to secure for the remainder of phase 1. I mentioned earlier that we're putting the governance structures in place. We've got terms of reference that we have drafted, and we're in the process of looking at the management group and whether and how we're going to expand that. We need to set up a new scientific advisory board and a patient public involvement steering group, and we will be recruiting members into that. So work is underway at the moment. We were obviously really delighted when we had the announcement a couple of weeks ago about the £4.75 million funding from the UK Government Department of Health and Social Care. That's enabling us to undertake the sequencing, but there's a lot of work that needs to go around that. So we were delighted when the We and ME Foundation put some money in to help fund some of that activity. But there is more work that needs to be done. And we, alongside the sequencing, we need to be able to undertake analysis, etc. And we've got the further phases that we need to put in place. So your support, as always, is critical. Please do help us by reading study updates, sharing information between community groups, your families and friends. Spread the word about sequence ME and long COVID. Some of you may have ideas around fundraising or have access to people that might be interested in becoming a strategic partner or funding the study. We also want to reach out into the long COVID community. We have a lot of relationships there already as an organisation, and we know many of you do, but at the right time, we'll be seeking expressions of interest in readiness for recruitment. And of course, as some of you already have done already, you can donate to Sequence ME and long COVID, and we've got the website address there for you as well. You can contribute directly into the study or to our work more broadly. One thing I would like to highlight before I hand back over to Chris is that this study is a result of a lot of work, not just the work that was undertaken with Decode ME, but also the years and years of advocacy that went before that. The work through what was the research collaborative that was chaired by Professor Sir Stephen Holgate, and a large number of people involved, and other charities and organisations. And many of us have been advocating for years to get to this point. So it felt really important to acknowledge and honour the part that others have played. So I'd now like to hand back to Chris to say a little bit more about phase 2.
Thank you. So phase 2 is the delivery of the data, that is to the generation of data of the whole genomes of 6,000 people with ME in the Decode ME sample collection. And that has been made possible by this fantastic award that we have from the UK government. If you go back, because I want to acknowledge, thank you. I want to acknowledge the contribution made by the We and ME charity, because without their contribution, we would not have been able to start work this year to employ the people who are generating the data, because the UK government's award didn't cover that. The other thing to say from this slide is that the control data, from the population controls, that data is already available via the NIHR bioresource, and we've had good conversations with them, for which I would like to thank them. Next slide. Thank you. So we're doing a type of sequencing called nanopore sequencing that is different from the more commonly used short read whole genome sequencing. So I wanted to say something about that. And that is a technology developed by Oxford Nanopore Technologies, and it involves this pore that is inserted into a lipid bilayer, and there are many, many such pores. And so at the same time, different strands of DNA, long strands, can be fed through these pores, and that leads to the sequencing of each of these long strands at the same time. The sequencing is done because there is an electrical voltage change as the DNA passes through the pore, and that change is different for each type of sequence, each letter difference as it passes. And it's that change in voltage that is read out, and from that the sequence is fairly accurately inferred. And we're not going to sequence each genome once because that would have some inaccuracies. We're going to do it 30 times on average for each person. Next slide. So this type of sequencing, this long read whole genome sequencing, it does many things. It will be able to make visible the common variation that we talked about that was seen in Decode-ME, but it makes visible also the rare genetic differences that people have in their genomes that are often just shared with people from your own extended family. And so we can start asking questions about that rare variation that we could never do so before. Like, is there a cluster of these rare variants that just disrupt the same single gene again and again? And are there explanations of people's symptoms that actually don't relate to ME at all, but to other conditions? And if so, we might be able to see those rare genetic differences that explain parts of your symptom profile. Then there are these larger parts of our genome that might differ in terms of having one more copy or one fewer copy. They might be inverted as a piece of DNA that might have jumped from one chromosome to another. And we'll be able to see those that the previous technology could not. And then lastly, a change to what we've previously done in Decode-ME, we're measuring what methylation occurs on DNA, and sometimes called the fifth letter, the fifth base of DNA methylation. And that measurement of DNA methylation might yet be shown in some way to be associated with people's ME symptoms. Next slide. We have done a pilot study now quite a long time ago, in the last quarter of 2024, and with 10 samples, we were able to show that this technology does work. It gives highly consistent results. We're able to have good scalability of the samples and the data. And so that convinced us that this was going to be possible, and we used that evidence in our funding application. Next. So just to say again, this project, the whole genome sequencing for ME and long COVID, is a Decode-ME project. That means that when you have given us your consent, your data will be available to other researchers as before, so that others can use it for their research and again to accelerate research towards effective treatments. And again, patient and public involvement, as it has been from the beginning, as Sonya has said, is going to be a part of this project, not just at the beginning and the middle, but right up until the very end. And the next slide. So this is just again who is currently working on the project and who is funding the project. Thanks to Oxford Nanopore. And I should also shout out to the European Bioinformatics Institute, EBI, and the group of you and Bernie, who are ready and waiting to receive the data. And just to remind you that the existing funds that we have received are not to analyze the data, but to generate the data. And so subsequent portion of this project will be undertaken by Ewan and his team at the EBI with funds that we have yet to secure. Thank you.
Okay, thank you, Chris. So we've just put a reminder up in the chat. There is a Q&A channel, so please do pop your questions in there. I'm just going to ask our co-panelists to introduce themselves. So maybe Andy, if you'd like to introduce yourself before we move to Hannah.
Yeah. Hi, everyone. If you don't know me already, I'm Andy Devereux-Cook, patient of 40 years, co-founded the Science for ME online forum, and I've been member of the management team and the PPI steering group at Decode-ME right from the start of right from the official start of the project.
Thanks, Andy. And Hannah?
Hi, good afternoon. I joined Action for ME a couple of weeks ago as the Business Development and Partnerships Manager, and I'll be working with the partnership to help raise funds and secure investment for the next phases of the project. So this is a 2-year post, so there's a lot of work to be done. Thank you,
Thanks,
Chris.
Hannah. So we have a few questions in the chat already, which I will share out to panelists. If we have too many questions, which may be the case, then we will take them away and we will, when we can, update the frequently asked questions. As I mentioned before, that's on the Action for ME website, on the pages specifically for Sequence ME and Long COVID. We'll also be making the recording of today's webinar available over the next couple of weeks. Just bear with us. We're a little bit stretched in terms of capacity, but that will be made live and we will make sure that we push that out through social media as well. So I'm going to direct the first few questions to you, Chris. You've already told us about the Decode ME samples and the fact that we're reusing them and that scientists can apply to access the genetic data for their own research. Can you just remind us of the process that we go through in Decode ME with the Data Access Committee, just so that people know what's involved and what we do to make sure that data is going to be used in the way it was intended?
Thank you. Yes. So there's an application form that is on our website at the University of Edinburgh, Decode ME, that people complete. And it has various sections, including, what are the questions that are being asked, what type of data samples are required for their work, what are their plans for PPI. And once that is received by us, then we discuss that application at the Data Access Committee, in which there are 2 independent members. And we most often, I think, we come back with some suggestions or questions for the proposers, and then they need to come back to us with their response. And if successful, then there's a phase when we draw up a transfer agreement, a contract as to how we're going to pass over the data. We have to be entirely certain that they're going to have very robust data security in place. And once that is understood to be the true, then we will then pass the data over. And that is for a period of time, after which they have to destroy the data.
Thank you, Chris. And those decisions around the data sharing are undertaken by the management group. So it's myself, Chris, and Andy with PPI embedded within it. Chris, the 6,000 samples that are going to be sequenced from Decode ME in phase 2, were they selected randomly?
They're not going to be selected randomly. There are some criteria that we are putting in place. The first is that the sample quality is high, and we have to ensure that we use the money wisely, and so we sequence those samples that are the best quality. We are prioritizing people who are most severely affected with ME symptoms, and we're doing this because we think it's the best use of the funds, but also scientifically we have the most expectation that they will have rare damaging variants to genes relative to people who are more mildly affected. We are what other criteria are we using? We're ensuring that people have given their consent for their data to be shared at this current point in time, so that when the whole genome sequence data is available, it will then be passed on. I think that's it for now. Yeah, I think so.
Right, and I'm going to ask this to all the panelists. I'm going to start with you, Andy. What are you most excited about with this project?
Well, apart from the obvious answer, continuing to work with Sonya and Chris, and challenging them in that way. I think it's continuing to build on the achievement that Decode ME was, that we've got the opportunity to take that achievement and take the next step or several steps forward from that. Because unfortunately, so often in the past with ME/CFS research, it does seem to be the case that the study is done, highlights something of potential interest, and either the research team does not continue to work on that, or to be fair, they may not have been able to secure funding for that, or for whatever reason their interest goes elsewhere. So I think the fact that we're able to essentially keep our core team together, keep our focus pointed forward, and the ability, as I say, to build on what we have achieved, and looking forward, with the hope and expectation that we'll be able to achieve so much more in the future.
Thank you, Andy. Hannah, it feels slightly unfair because you only joined us a couple of weeks ago, but I'm going to ask you anyway. What about you?
Well, I'm excited just to be in this job, so as a first. But yeah, I think just adding to what Andy said, to be part of a groundbreaking landmark study, I think is hugely exciting. And from a fundraising and seeking investment perspective, I'm excited about broadening the pool of funders that are then, I hope, going to be advocates and lobbyists for more investment and interest into ME and long COVID for the long term. Thanks.
Thank you, Hannah. Chris, what about you?
We need to hear from you as well, Sonia.
Yeah, I'll go last.
So I think the exciting thing that we're on the verge of is getting answers. That ME/CFS is a disease with too many questions and too many potential answers that people have floated for a long time without there being really firmed up, replicated evidence. And I think with this project, we're on the verge of being able to give answers, not for everyone, but for some people. And once those answers come about, then they will definitely help in answering all of the other questions for everyone else.
Thank you, Chris. And I will answer that as well. I'm not going to, I agree with everything that everybody said, so I'm going to say something slightly different. But it does build on what Chris said. I think, many of us have waited for a very long time for answers. And I know several of you in the audience will have been even longer, many decades. But we have to dispel the myths that are out there. We have to silence those individuals that actually have done damage to this field and to this community. And I think we have shown through this process, as I mentioned, the advocacy work, the collaborations, that we can accelerate research. It's not near enough and it's not fast enough, but things are starting to move. And I think I'm excited about being part of a team that is, and not just the team in front of you at the moment, but a wider team, really committed and driven to make sure that that happens and that it happens more quickly. So I think this is, feels like an important point, but we also have more to do. So Chris, I'm going to come back to you. I'm going to ask you two questions. They're totally unconnected, but it'll give you a chance to answer both. Is AI something that could help accelerate this project? And then completely different, is there any genetic correlation between ME, long COVID, and autism?
Taking the second one first, we have not looked at that, but it is one of the things that we will be reporting on in the updated version of the preprint, which we will send to a journal. So I pledge that you will know the answer to that at that point. In essence, on AI, we are already using AI in terms of big data science. This is already in play, and we pass the data over to others such as Precision Life, who are also using different flavors of AI. There are many bad things about AI, but we're obviously trying to use it for good.
Thank you. And what's the timescale, Chris, on getting some initial results to the public and
Yeah.
for completing the entire project?
Yeah, I can't give a timescale, and the reason is that we don't have any funds whatsoever to do the analysis. So we will generate the data this year by March 2027, but we don't have the funds that we will need to do the analysis. If we had those funds in-house in our back pockets today, I'd be able to give you that timeline. So apologies, I can't answer that.
Thank you. And another question that's come in is somebody's asked, are they right in thinking it's about 15 million that's needed to complete the study? It's around that. Costs are changing all the time, given the current sort of financial and economic world climate at the moment, and therefore our figures keep changing. We are anticipating this is a £20 million study. And one of the things that Hannah's going to be doing, I'm not going to put Hannah on the spot at the moment, is working with us to look at costing for each of the subsequent phases. But as I said, each time we do that, the figures change. And so we are working on the basis that we've got another 15 million to fundraise against. We will be looking at what money might be available through applications, but we are also engaging with a number of partners like the Schmidt Initiative for Long COVID, CODA, Sequence ME, sorry, not Sequence ME, Solve ME, We and ME, and others. We know that we're going to need a whole group of people being partners on this study to help us fund this. And we have been very fortunate that we've had some individual donors as well. So we are looking more broadly. We know how important this study is to get moving and to get it moving as quickly as possible. And that's why being able to fund Hannah's role has been really critical within phase one. Chris, will the results for ME and long COVID be shown together or as two separate conditions? And I'm not going to read all of the detail of the question, but essentially the individual is really wanting to understand what difference we think Sequence ME will make, particularly to those who continue with a wrong understanding of portrayal of ME, and also, as we've seen, that psychologization with long COVID too.
So this is a question for the management group via PPI as to how we're going to deliver the results into the public domain. It's not my decision only. My personal point of view is that we should analyze the data for people with ME against the population controls and provide those results as soon as we can. That's the first answer. The second is that, I mean, I hear you when you said that we need to silence people, Sonya, but the way that I try and move the conversation on is not by silencing people, but by providing the evidence so that there is no response to that evidence. And I think that's what you meant anyway.
It is, yeah.
And that's where I think we're at, is that with further evidence of the genetic predisposition to ME, and other disorders, fibromyalgia, long COVID, etc. I think the idea that this is only in the mind is diminished, but it's not gone away. And what we're doing here is trying to work out not, whether it's a psychological condition or not, but what is going wrong? What at the molecular level? What at the cellular level? What does that mean? How does that relate to people's symptoms? Why is it that it's a female biased disease? Why is it often an infection at onset? We're trying to work out what are the answers to all of those questions using genetics, and that is a very tall order to do, but really I think only genetics can provide answers to these questions in time. I don't think it's going to be soon, because all of the other types of studies provide correlations and therefore can be interpreted in a variety of different ways, including by people who have different perspectives on the science than I do. And I would like to ensure that the interpretations are limited to ones that have substantial evidence for them.
Thanks, Chris. And you are absolutely right. It is, that evidence is absolutely critical. What we have heard as Action for ME from people that were able to take the Decode ME results into their doctor's surgery, even the next day, and be able to share the results. And that has made a difference in terms of their engagement and their support and their treatment. So, there's a whole spectrum of ways in which this study and what's come before it can impact for people. To give you a break, Chris, I'm going to answer this question because it's to Action for ME. Will Action for ME be working to achieve other collaborations given the success of our partnership with Edinburgh University to increase research in other areas of science? We want to, and we're committed to. We are a very small team delivering a whole range of things, including services to people with ME and ME-like symptoms and their families and professionals. And so we have to be quite focused in terms of what we're doing around the relationships and the collaborations that we're building up. But one of the really exciting things that was announced last year was the PRIME project, which is the funding from the Medical Research Council. It's strategic partnership funding, and Action for ME is working not only with Edinburgh University, but a whole range of other researchers and academic institutions on research in a number of different areas and not just in genetics. You can find out more again about that on the website. There are various workshops being run, and we have our first symposium that's going to be held, which actually is on genetics for this first one at the end of September, and that will be online and also in person at Edinburgh University. So we have to be focused. We can't do it all, but PRIME offers a real opportunity to broaden those relationships, and I think already it's starting to yield some progress in forging new collaborations. So Chris, the pilot that you told us about, the 10 samples that we did many, many months ago at the end of 2024, was that data published?
So the data is not published, and that because it's a pilot and not a fully fledged project. We can do and will do so much better with larger numbers of samples. And if the question is about publishing the data, we won't put the data into the public domain. We'll make it available to researchers, but we won't publish the data. But a sample of 10 individuals, when we don't have controls, would not have given us any highly supported evidence one way or the other on ME, so we wouldn't have published it.
Andy, question here. Will we find out if our Decode ME sample is one of the ones being used?
I'm pretty certain no. And I have to admit, that's something that's been decided on quite some time ago, so it's dropped off dropped out from my memory banks the exact answer, but I wouldn't expect so. Obviously, one of the key things about giving samples, particularly to these kind of projects, is anonymization, and it's almost the key thing that you won't know is exactly what's needed. Basically, because you can't know, nobody else can know the results. So your privacy is assured in that regard. So your genetic data is kept private and is only seen as essentially an amalgamation with all the other results. So as I can understand the temptation to know that your particular sample was part of Decode ME and the next step into sequence ME and long COVID, but unfortunately, no, we can't give out that kind of information.
Thanks, Andy. There's a number of questions here around sort of pressure and campaigning with the government, etc. We don't have one big national campaign. As a charity, Action for ME engages with our parliamentary champions, working with the all-party parliamentary group, etc., to really advocate and work collaboratively to encourage funding. And that work led to Decode ME and has led to the 4.75 million that was announced fairly recently. We recognise there are lots of challenges on the government. There are lots of calls on funding, etc. And we know that there is funding available for people to apply to. But we will continue to work in the way in which we have to try and accelerate ME and long COVID research. There are things that can be done, encouraging your MP to come along to all party parliamentary group meetings, writing to them to share your experience. As an organisation, we have a number of questions that we ask parliamentary champions to submit. We support MPs and peers to apply for debates, etc. We haven't undertaken what has been described or asked for a coordinated campaign in the questions, but there are a number of ways in which people can support that. And what we'll do is take away some thinking around whether or not it might be helpful to have a separate webinar around what do we do and what impact does it have, and we'll maybe pick up some of the questions there. Chris, I'm going to come back to you. Could you explain what the letters are in reality? And that was referring back to the letters in your earlier presentation.
So DNA folds up in this double helix, and as you walk around the helix, you will bump into 4 different chemicals. And instead of giving the long name, the scientists change the name into single letters, A and C and G and T. So in our communications, we wanted to make it as simple as possible. So that's what we do. We talk about those 4 letters. And when we add the 5th, which is the change of the C into a methyl C, we talk about that as the non-inherited part of the genome. The 4 letters are inherited and the methylation is not.
Just to jump in,
yep,
don't suppose Chris could expand on methylation a bit, because you mentioned that earlier in the presentation, but
it's a modification to one of the letters, the C letter, and I think it's a carbon and 3 hydrogens. If someone's going to tell me if I'm wrong, and that chemical modification has some implication as to the activity of the surrounding genes. And so people analyze it to ask questions about gene activity. And I see there are questions about the sample, because the sample in Decode ME is from saliva. And so the methylation is only relevant to the saliva sample given rather than any other portion of the body.
Great. Thank you. And just to build on that, there's a question here asking around about methylation analysis. Is there a chance to learn anything about post-exertional malaise from that?
I think not. I don't want to raise hopes on that. We don't know what people's PEM status was upon giving a sample, and we don't have serial measurements before, during, after, for example. That would be a different study. I'll tell you what I think, and that is the most useful part of the data will be the 4 letters, A, C, G, and T. We get the methylation for free, so we will analyze it, or someone will, but I am most interested in the 4 letters.
Thank you. There's a question on here around peer review for the Decode ME study results. And as Chris mentioned earlier on, we are working on a publication and we are looking for that to go into a peer-reviewed journal. We will also update the preprint that is already online, and work is being undertaken on that at the moment. So we will, of course, let you know when we're ready to announce that, Chris.
And people may have heard about the UK Biobank and their changing of their data security processes, which I support, obviously. But that has meant that they have shut us and everyone else out of their data analysis platform. And it's been, I think, a month or so now, and we don't yet know when we're back in. And that is causing us the delay at the moment.
Thank you. And linked with that question, if you can just explain, Chris, were all of those involved in the Decode ME study, did they have a firm diagnosis from a medical professional? I know we undertook a lot of work in terms of developing our algorithm to assess those people that applied, and we then recruited and invited DNA. Is there anything else you want to add to that?
Every person who was analysed for Decode ME GWAS had a medical diagnosis of ME from a health professional, ME, CFS, and the veracity of that self-report is demonstrated by the fact that we did see genetic signals. If that was not the case, that people were in some way making it up, we would not have seen the genetic signals.
Thank you, Chris. We're going to be wrapping up in a moment. There are a number of questions here around the recruitment for people with long COVID and those participants. As I mentioned earlier, we have yet to set the criteria. We will be working with a large number of people to help determine and define that. That includes long COVID charities. It will include people with lived experience of long COVID, as well as those people with lived experience of ME. We have yet to establish that process, but there are opportunities, particularly through the networks that we've built through the Prime project that we mentioned earlier on, but also the relationships charities like Action for ME have through the Overlapping Illness Alliance, etc. So we don't have a process determined for that. That's something we said we do within the 2 years of phase 1, and that work has yet to start. But as we have done with Decode ME, we will be transparent, and we will keep you updated on our progress. This is the first of what I anticipate to be a series of webinars. So on that note, I'd just like to say, first of all, thank you very much to my co-panelists and to Chris for your presentation, but also to Daphne and other members of the Action for ME team who've worked in the background and helped in a whole range of number of ways. As I said, the recording will be live. It is going to take us a little bit of time. We will let you know when it goes live. You will be able to access that freely. We will update the website and the frequently asked questions when we can, so do keep an eye on that. And that will include letting you know when we open for expressions of interest for the long COVID recruitment piece. So thank you all very much for joining us. We hope to be able to have some more exciting updates in the not too distant future. But just before we go, I'd also like to acknowledge our other partners, Oxford Nanopore Technologies, and our funding partners, and also Embl EBI, who'll be working with us in the future. Thanks very much, and hopefully see you all soon. Bye.
Thank you, everyone.